Okay, so this video is just to run you through some of the theory behind the pGlo experiment that we're doing in Units 3, 4 and Biology this year. You may use this as part of your revision, watch, rewind, and make sure that you understand the content, the process and the method that we've used, and some of the risks involved in the actual process. Okay, so some of the accuracy contamination risks that you might be asked to discuss, and also be able to describe some of the considerations when we are producing a genetically modified organism, which is what this experiment's all about. So we call this pGlo bacterial transformation. pGlo is the name of the plasmid that we use, and this plasmid has been engineered already for us. So they've used a gene from a jellyfish that will fluoresce under your ultraviolet light, and they've taken that gene, and the gene we call GFP for green fluorescent protein, and they've added that gene into a plasmid that can be incorporated into bacteria, and we'll be using a non-pathogenic E. coli in this experiment. So here we've got a slide showing you the jellyfish, Aquaria victoria, which expresses, as part of its natural genome, it has the gene for the GFP, and when that gene is expressed, there's a part of that GFP protein will fluoresce under UV light, and what you will observe, the phenotype that you're looking for to see if this gene is being expressed, is a greeny glow. Okay, so a yellowish green glow when we shine a UV light on it the day after the experiment. So just some key ideas that you need to be familiar with. You need to understand that the mechanism that we're using to transform the organism. So we're using a single-celled organism. So bacteria in their single-cell form are a whole organism themselves, and we're going to transform that organism. The advantages of using bacteria are that bacteria have a very short life cycle, and we can introduce plasmids quite easily that carry the gene. So the plasmid is the vector in this situation, and we can take that, we can modify the vector, and we can introduce the vector to the cell, and then the bacterial cell, the bacterial organism will express the new genotype that we've introduced. So genetic transformation is the process. Gene regulation is a really important concept within this experiment. So I recommend that you look back at your lac operon and how genes are regulated in prokaryotic cells, remembering that the operon is a series of genes under the control of one single promoter region and has an operator region which is used as the switch, and you need to be able to describe the role of regulatory genes that will be found upstream of the structural genes. And also genetic selection. So how are we going to identify the organisms that have taken up the recombinant plasmid from those that haven't? Okay, so quick content refresher. DNA is the genetic code that holds the genes. The transcription of DNA to RNA occurs once RNA polymerase binds to the promoter region and can actually collect free nucleotides and build an RNA copy, which will then be translated at the ribosome into a polypeptide chain and a final protein. And that protein will give the organism its traits. Okay. So this is the process I've just outlined. It's a really, we call it the central dogma of biology. And it's the idea that it's the whole process of gene expression. So how we go from genes and DNA to the protein and the final phenotype. So that's a really good idea to refresh your memory on the process there. And remembering that what I'm showing you here is eukaryotic transcription and translation. So we've got a nucleus here which tells you that it's not a prokaryotic cell. Whereas today, or in the pGlo experiment, you'll be using bacterial cells. So they don't have that nucleus, and transcription and translation can happen almost at the same time. So this is a stylized drawing of the bacterial cell that we'll be using. Now remember that the bacteria have a single chromosome, a loop within the cell, and that holds most of the genetic information. But these tiny loops are called plasmids that you can see scattered around the cell here. And what they can do is they can enter and leave the cell. And we're going to try and encourage the recombinant, so the genetically engineered plasmid that has got the GFP gene, incorporated into it, we're going to try and encourage that to get inside the cell, and then the E. coli will express the traits. So plasmids, a couple of things you need to know about plasmids. Yes, they're excellent vectors for transforming bacterial cells, and they're also, they can replicate outside of the cell itself. So yes, they will be copied as the cell divides into two, but they can also be copied within the cell. Antibiotic resistance is a very common gene to have on a plasmid, and we'll be using that today to determine whether or not the organism has taken up the plasmid. So refresh your memory on restriction enzymes and ligase. So restriction enzymes can cut DNA, and they can cut leaving sticky ends or blunt ends. And that's what the Biorad firm have done for us. So they've already incorporated the GFP gene into the plasmid, which is why we're calling it a recombinant plasmid. And this is the process that they've used to do that. They've used a restriction enzyme to cut both the plasmid and the gene of interest and combine those, ensuring that the sticky ends complement each other so that the GFP gene can be incorporated into the plasmid. Excuse the bell. And this is the plasmid that we've purchased. So this is the point of origin of DNA replication. Okay, that's not a huge part of today's content, but it is often labelled on the plasmid, so it's useful to know what it's there for. The BLA gene is the gene that provides antibiotic resistance, so we can use that to select whether or not the organism has taken up the plasmid by trying to grow the E. coli on antibiotics. If they can grow on antibiotics, we know that they have this gene incorporated into their genotype. And if they can't, if they're eliminated by antibiotics, then we know that they haven't taken up the plasmid. The RSC is a regulatory gene. It produces a protein that is the switch that turns on and off the expression of this structural gene, the GFP here. So this produces a protein called the RSC protein that binds to the operator region and then will block RNA polymerase from transcribing GFP unless we add an inducer molecule. And in this situation, it's arabinose is the inducer molecule, not lactose like in your lacoperon, but arabinose. And that changes the conformation of the RSC protein, removing it from the operator, or at least exposing the promoter region, and allowing RNA polymerase to transcribe, and then the E. coli to translate the GFP gene. So in this situation, what we're going to do is express the antibiotic resistance gene to produce an enzyme called beta-lactamase, and the A's here tells you that this is an enzyme, and it actually breaks down the antibiotic. So if beta-lactamase or the BLA gene has been incorporated into the cell, what you'll see is that ampicillin won't have an effect on the growth of the E. coli because it's producing this protein that breaks down the ampicillin. So that's our key marker to see if the recombinant plasmid has been taken up. So this is a diagram to show you the arabinose operon. So it's very similar to the lacoperon. If you remember, lacoperon relies on the presence or absence of lactose to determine gene expression. Here we've got the presence or absence of the arabinose. And the word operon means that we've got a series of genes under the control of a particular promoter. Okay, so we've got 1, 2, 3 genes here that in nature would be expressed to produce enzymes that break down arabinose. Now what Biorad have done have taken those genes for those enzymes away and replaced it with this GFP jellyfish gene. Now keep in mind that that means that we've now produced a plasmid from 2 different organisms' genotypes, so we've got a jellyfish and the E. coli. And that means that our final organism will be both a GMO, so modified, and a transgenic, because we're combining genes from different organisms. So what will happen is, in the absence of arabinose, the RSC protein will block RNA polymerase from transcribing this gene, and therefore it will not fluoresce under UV light. But as soon as we add arabinose, RNA polymerase, this blue circle here, will be able to bind to the promoter and express that gene and produce the GFP protein. And it's the protein and not the gene that will fluoresce under UV light. So the presence of the gene won't allow us to see it glowing in that yellowy greeny colour under UV. But if we can turn on expression of that gene and produce the GFP protein, then we will see that phenotype. So keep in mind, it's a really important idea, and lots of students get confused here. The gene itself, the GFP gene, does not glow under UV, but the protein that's produced when that gene is expressed will do. So, this is just a little diagram to show you what we're hoping will happen. If we can get the plasmids, the recombinant plasmids, to go inside the cell, then they will be able to, the genes in those plasmids for antibiotic resistance and the GFP will be expressed, and that will give us two different things to observe. The first thing, the selective factor, is will it grow on ampicillin, showing us that it's expressing that antibiotic resistance gene, and if we add arabinose, can we get GFP to express? So methodologically, so you need to understand the basics of how we're going to try and encourage the plasmid to enter the cell. We're going to use this second protocol called calcium chloride and heat shock. And in short, the calcium chloride is there to neutralize the negative charge of the DNA and stop the DNA from being repelled from the plasma membrane. So this is a flashback to your plasma membrane learning, where you know that things that are charged can't enter the cell. So we need to neutralize that negative charge first. And then what we do is we change the fluidity and permeability of the membrane, cooling it down, stabilizing that membrane, slowing down the metabolic processes, and then increasing the temperature and fluidity to encourage the plasmid to enter the cell, and then providing the E. coli with some nutrients so that it can then start to express the genes. So liquid broth, which you'll see is in all of the dishes, is really just a combination of the E. coli's favourite or necessary carbohydrates, amino acids, and things that drive its biological processes. So liquid broth is the ideal conditions for this E. coli to grow. And then what we will do is we will add the plasmid to only the experimental dishes, not allow the plasmid to enter the control dishes, so that we can actually draw some conclusions based on our observations. So if you look at the 4 types of dishes that you will be inoculating today, you'll have 2 with wild type pGlo. So we call the natural version of pGlo wild type, and this has not been exposed to the plasmid. So what you'll do is you'll grow some natural pGlo following heat shock on some liquid broth, and that will determine whether or not the bacteria has survived the heat shock protocol. Then what you'll attempt to do is grow some wild type, so non-transformed E. coli, on ampicillin, and what that's there to demonstrate is that the wild type or the natural E. coli are not resistant naturally to ampicillin. Okay, so they're susceptible. Then your experimental groups are here to demonstrate that if we can get the pGlo plasmid to enter the cell, we will have some colonies that are able to grow on ampicillin. But you'd expect that there are a lower number of colonies here than in your first dish, because the transformation rate is probably only around 5 to 10%. So you'd see less growth, but nevertheless any of those colonies that can grow in the presence of ampicillin are expressing that resistance gene and therefore have been transformed. And then your final dish has the addition of arabinose, remembering that that's the inducer molecule, and that's there to induce expression of the GFP gene by removing the repressor from the operator region. And if you have successfully transformed any of the E. coli, in the presence of arabinose, you will be able to observe fluorescence under UV light, showing that that GFP gene has been turned to express and turned into a GFP protein. But again, you'll have a similarly low number of bacteria when compared to dish 1 here. And you're expecting about the same number of colonies in these 2 dishes. Okay? This is an overview of the protocol which you've got in your lab manual. I recommend that you read through that and underline some of the key pressure points, because there are some really important ideas such as timing and temperature control. The risks of contamination are quite significant in getting accurate outcomes. So read through that, annotate it carefully, and you're advised to watch this video again if you feel you need to. Well done.